we're going to look at are the foraminifera. And so the foraminifera, their name describes them. Foram means hole or little hole, and fera to bear. When we look at the body of foraminiferans, and particularly this individual here, you can see all those little holes that are born across the surface of the organism. The members of the foraminifera are going to have a chambered calcium test, and a test is kind of like an exoskeleton or a shell, if you will. And so their test or skeleton is made out of calcium, similar to your bone structure, and it's going to be in these small consecutive chambers. And so it ends up looking like a snail shell or a leaf or even like the button on a rattlesnake. But that's one way you can tell you're looking at the foraminifera. These organisms are primarily marine, and this chambered skeleton, as these organisms die, okay, so as they grow, they start out here in the middle, and then they get bigger, too big, so they secrete a new chamber for themselves, and they keep growing, get bigger, 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 bigger. Well, eventually, they die, and when they die, these calcium tests sink to the bottom of the ocean, and they form the sediment. Over time, that sediment accumulates, and this is where we get one of our products we use today, limestone. And so limestone, portions of it are made of the calcium for the foraminiferans. Limestone was what was used to build structures like the pyramids, and we also use it here in Texas to put on the front of our houses. And if you've ever driven down through the hill country, you'll see the road goes through these huge white hills. Well, that's because you're driving through what used to be the bottom of the ocean. And if you were to take a slide and prepare it, take a sample and prepare a slide from it of that material, you would most likely find the tests of these foraminiferans left over. And so they're an interesting group. Our next group in the rhizaria are the radiolaria. The radiolaria are all going to have radially symmetrical tests composed of silica. And so they look like they have little shells of glass or exoskeletons of glass that can be divided or that are in perfect circles. When we look at the radiolarians, about 90% of all described species within this group are extinct. And so they're great for geologic dating, helping us understand, okay, in this particular strata or level of sediment, we find these radiolarians with this particular fossil. By understanding at what point that radiolaria lived, we now know at what point that fossil was formed or when that organism died in that particular sediment. So they're very important for geologic dating. Our last supergroup is supergroup alveolata. In this group, all members are going to possess flattened vesicles called alveoli. These are stacked in a continuous layer between the plasma membrane, and so here you can see them, these little kind of bubble-looking structures beneath the surface. And in this paramecium, you can see them along the edges. That's what's going to help give the structures or give the organisms their shape, and so this is a unique feature for the alveolata. They all have alveoli or small sacs or vesicles underneath the plasma membrane. There's three subgroups: the dinoflagellates, the apicomplexans, and the ciliates that we'll be discussing. And so we'll begin with the dinoflagellates. Dinoflagellates get their name from the Greek word dynon, which means to whirl, They are going to be planktonic organisms that float or drift through the ocean. Now, that doesn't mean they're not moving at all. These organisms possess two flagella, and they're going to lie in grooves that are perpendicular to each other, meaning one goes this direction, the other goes this direction. The movement of the flagella is going to cause the dinoflagellate to look like it's whirling or spinning through the water. Now, these particular organisms are commercially important because they cause the red tide phenomenon. And red tide occurs when there are periodic blooms in dinoflagellate populations, and that results in the accumulation of a toxin that they produce as part of their metabolism in the water. So here what you're looking at, this is red tide. So what you're seeing is that there's a pigment within the dinoflagellate that's causing this coloration, and the toxin they produce is staining the water. As that toxin accumulates, which normally there's not so many of them, so it dilutes in the ocean and it's not a big deal. But whenever you have these blooms where you have hundreds of millions of trillions of individuals all producing this toxin, you surpass what's called, that normal threshold amount. And now fish start to die, lots of marine organisms die, including shellfish. But the problem is that we like to eat shellfish, so think about things like clams and mussels. And the problem is that whenever you harvest them, you can't really tell if they were dead before or they're dead after you pull them open and harvest the mussel itself. And so we've seen people actually die from the toxin produced from dinoflagellates after consuming shellfish. So just be aware as you're traveling, look for any notifications about red tide and maybe stay away from the oysters. All right, our next group are the apicomplexans. Their name refers to their structure. So api comes from apex or tip. Complex is from this twisted around structure found at the tip. This is important because these organisms are important human parasites. And so that's what they're going to use to enter human cells and complete their life cycle. So they're all parasitic, living inside of some other animal. They all possess an apical complex, this structure, and it kind of works like a drill bit to work their way inside the cell they're going to parasitize. This particular organism, Plasmodium is the one we're going to focus on, causes malaria. And so Plasmodium is seen here inside the red blood cell, that little yellow guy right there, and so that one's actually ruptured. You can see this one that's just about to rupture. That is the Plasmodium individual, and so Plasmodium is a genus. We're looking at just the group in general. Right? So this particular organism causes malaria. It's going to be found inside red blood cells, and this is a huge distinction whenever we compare it to Trypanosoma. Trypanosoma was found in the fluid around red blood cells. All right, so let's talk more about malaria. There are 3 to 5 species within Plasmodium that cause malaria. We're just going to talk about generally how malaria occurs. Malaria is spread from one infected host to another by the vector, the Anopheles mosquito, and specifically it's the female Anopheles mosquito because she's the only one that actually feeds during the life cycle. Males do not eat once they hatch out into the adult form. All they're worried about is reproducing with females. And so the female takes a blood meal from an infected host. We're going to see a life cycle that includes division within the gut of the vector, and the vector, while not suffering any detrimental effects from that infection, will carry the parasite to the next host for infection. And so here we see how that infections occur. As an individual becomes infected, so here we see the little green cell, that's the parasite that's moving from the salivary glands of the mosquito into your body. Those parasites are going to make their way through your bloodstream, so this is your capillary, through your blood to your liver, to a specific type of cell called a hepatocyte in the liver. Once in the liver, they're going to begin to divide and divide and divide. Eventually, you get so many that the cell ruptures. Well, when that cell ruptures, your hepatocyte rupture, it's going to spill all of those protists into your circulatory system. And so now those protists, the Plasmodium, are looking for red blood cells to infect. As they encounter red blood cells, they're going to use that apical complex to drill inside of it, and then they're going to undergo a process of division while inside the red blood cell. Divide, 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 until eventually you get so many inside the red blood cell that it causes lysis or rupturing of the red blood cell, spilling those organisms back out into the bloodstream. And so this is why we see the periods of chills and fever associated with malaria. You get fever as these cells are rupturing, spilling that parasite out into your circulatory system as a whole. Your body, sensing that foreign invader, ramps up temperature to try to kill them off, but then remember, as each of these parasites is being loosed, it's looking for a new red blood cell to infect. You have millions of red blood cells, and so they're encountering these blood cells frequently, making their way inside. And so after a little bit, even, and it's a period of hours, but after about an hour, most of those individuals have either reinfected a red blood cell or a new red blood cell, or they've been killed by your immune system. And so your immune system now has nothing that it can recognize to fight because everything is housed inside a cell that belongs to the body. And so you're going to see the fever drop off, the chills as a result, until eventually new cells rupture and it all starts over again. So you see this periodic up and down of fever chills corresponding to this lysis that's occurring in the cell. Now, if an individual that has been infected by Plasmodium is fed upon by another female Anopheles mosquito, we see that she will take up the parasite itself, and now you can see the division that's going on in her body, getting it ready to go back to the salivary glands, and then she will pass it on to a new individual. All right, never going to ask you to explain the life cycle to me. I just want you to understand how it works. This is an important slide for you to use while you're studying because it's going to allow you to compare the difference between Plasmodium and Trypanosoma. So Plasmodium is an alveolate, Trypanosoma is a euglenozoan. Remember, euglenozoans have that flagella that is prominent and visible, and they live in the fluid outside the red blood cell. When we look at Plasmodium, they too have a flagellum, but you're never going to be able to see it really clearly. This is about the best I can do. It's short and it's very thick. Plasmodium is going to infect the red blood cell's interior. And so that's an important distinction that's going to allow you, that's going to allow you to be able to differentiate between them. So inside the red blood cell is Plasmodium, outside the red blood cell, Trypanosoma. All right. Our last group that we're going to talk about are the ciliates. The ciliates get their name from their locomotion structure, and so cilium means eyelash for to bear. When we look at the body of ciliates, we see that they are covered in small hair-like extension called cilia, and that's what they're going to use to move. And they beat like oars on a ship or a boat, or like your hands would if you were doing the breaststroke, to pull the body of the protist through the water. Now, most of our ciliates are going to be free-living, non-parasitic form. They differ from other protists in that they have two nuclei, and this is an important feature. They have a macronucleus, macro means big. They have a larger nucleus that's important for metabolic function, and it controls the metabolism of the organism. The micronucleus is important for sexual reproduction. We're going to see that when ciliates conjugate, or they're going to exchange their genetic information, the micronucleus is what's going to dissolve and allow that to happen. The organism that we're going to focus on from the ciliate group is paramecium. And so here you see a paramecium covered with its little hairs all over. Isn't he precious? And so the paramecium are going to use a prominent oral groove for feeding. Here, cilia are going to beat in a to create a vortex, if you will, that's going to pull food molecules in from this direction into this feeding groove or oral groove, and a food vacuole would form on the other side. And so that's how they're going to take in small protists and bacteria that they encounter in their environment. As paramecium reproduce, there's two ways that they can do this. The first is through binary fission or splitting in two. And so here we start with one parent paramecium. After it receives an appropriate signal, it's going to split all of its organelles, divide in half, generating two genetically identical offspring. Sexual reproduction in the paramecium occurs through conjugation. And so here, conjugation is going to allow for two paramecium to attach to one another. The micronucleus degenerates, and they will swap segments of their DNA to allow for variation in the population. As they're conjugating, so they get together and they shake and they're exchanging DNA, and then eventually they separate, and then they'll undergo binary fission to generate new offspring. All right, guys, so that's the end of our lecture today. I hope that helps you as you're studying and just helps you fill in the notes that we were supposed to cover today. And again, to those of you that showed up to class, my apologies for that you didn't get the email sooner, but just use this information to help you keep up with what we're doing. And then next time we will go through all of the lab material pretty thoroughly. So if you have any questions, please email me. Otherwise, we are done. Have a great day.